Hi there, in this video we are going to discuss various cellular adaptation to stress. When cell is exposed to some stressful environment, it tries to change its number, its size, its metabolic pathways to adapt to the stressful environment. These cellular adaptation can be physiological adaptation and pathological adaptations. Physiological adaptations are a normal response to the stimuli, stressful stimuli, such as increased in hormones or increased in some growth factors, and these adaptation help cell to perform its function in a better way. While pathological adaptation are those which are towards the damaging environment and cell try to escape the injury, and these adaptations are pathological and they are attained on the compromise of cellular function. So major cellular adaptation which we are going to discuss here are hypertrophy, atrophy, hyperplasia and metaplasia. So first is hypertrophy. What is hypertrophy? Hypertrophy is increase in size of cell during increase in size of cell, which leads to the increase in size of entire organ. So hyperplasia is increase in number of cells. We need to differentiate between these two terminologies here. Hypertrophy is increase in size of cell, while hyperplasia is increase in number of cell. So hypertrophy occurs in both state, in physiological hypertrophy and pathological hypertrophy. Both are present and both occur in human body. So when there is physiological hypertrophy occurs during the enlargement of uterus during pregnancy, which is mainly induced by enlargement of smooth muscle cells and increasing number of smooth muscle cells of the uterus. So when there is enlargement of uterus during pregnancy, there is both hypertrophy and hyperplasia. While when skeletal muscle are enlarged as a result of strenuous exercise, this is mainly because of only hypertrophy, because skeletal muscle has limited capacity to divide. So hypertrophy is mainly a predominant feature of those cells which have limited ability or limited capacity to divide, while those cells which can divide are mainly they mainly adapt to stressful environment by increasing their number or hyperplasia. This is the example of physiological hypertrophy. Physiological hypertrophy occurs during the enlargement of uterus during pregnancy and enlargement of muscle fibers during the as a response to strenuous exercise. While pathological hypertrophy occurs in the cardiac muscle cell when they are exposed to hypertension, chronic hypertension, or aortic valve disease. When there is chronic hypertension or aortic valve disease, myocardial cell have to perform additional function or additional work. There is a workload on this cell to work to pump blood against this increased blood pressure or aortic stenosis, and when they perform more work, they undergo hypertrophy. And when they undergo hypertrophy, their contractile force increases and they pump blood better in a better way against this increased blood pressure and aortic stenosis. But there is a limited capacity to this hypertrophy. There is a limited capacity to which cell can increase its function by hypertrophy. Then a stage reaches at which hypertrophy cannot increase the cellular function. When this stage is reached, there is myocardial fiber lead to the degenerative changes which lead to the cardiac dilation, and when the chamber of heart dilate, there is failure of the cardiac function and ultimately leads to heart failure. So what induces these cardiac hypertrophy? Mainly there are two type of stimuli which lead to the cardiac hypertrophy. These are mechanical triggers and soluble mediators. Mechanical triggers, what are mechanical triggers? These mechanical triggers basically are increased workload, and when there is increased workload, various soluble mediators and various chemicals, growth factors and adrenergic hormones act on the myocardial cell, and they also lead to the signal transduction pathway which lead to the transcription of genes, and these genes lead to the production of contractile proteins, and these proteins lead to the formation of new myofibrils and myofilaments. As a result, the contractile force of cell increases. There is also shift from adult protein to fetal or neonatal contractile proteins, which have economical contraction. It means that they contract well, but energy expenditure is low on their contraction. So this is basically cardiac hypertrophy is basically a pathological hypertrophy in response to hypertension or aortic stenosis, and it improves the cardiac contractile force to a limit. When this limit is crossed, there is no further increase in the contractile force, and it will ultimately lead to the cardiac dilation and cardiac failure. So why there is a limit to these hypertrophy? It is because there is a limit to which vasculature or blood supply can blood flow can make nutrients available to the hypertrophied muscles. So there is also limitation of blood supply. Then there is limitation on the mitochondrial function, because mitochondria can also supply a limited amount of ATP, and there is also limited biosynthetic machinery to provide sufficient proteins and cytoskeleton elements. So these limited supply of blood, limited supply of ATP by mitochondria, limited supply of protein by biosynthetic machinery, and limited supply of cytoskeleton materials all collectively contribute to this limiting factor, limiting ability of cell to hypertrophy. So whatever physiological hypertrophy, was in uterus and smooth muscle of uterus and skeletal muscle, and pathological hypertrophy occurs in heart muscle. These are some examples of hypertrophy. Next we come towards hyperplasia. So hyperplasia is an increase in number of cell. It is not in enlargement in size of cell, but it is increase in number of cell. So this increase in number can occur in both way, by division of fully differentiated cell and by increase in number from the progenitor cells. And then similarly, as was hypertrophy, hyperplasia can also be physiological as well as pathological. So physiological hyperplasia is also of two types. One is hormonal hyperplasia and other is compensatory hyperplasia. This hormonal hyperplasia is an increase in number of cells in response to hormonal stimuli. For example, glandular epithelium of the female breast increase at puberty as well as during pregnancy due to hormonal impact. This is hormonal hyperplasia. And what is compensatory hyperplasia? Compensatory hyperplasia occurs to compensate the lost part of an organ. For example, when there is liver transplant, part of liver is removed from the donor, and the remaining liver regenerates its lost part in donor by hyperplasia. This is compensatory hyperplasia. So these are the example of physiological hyperplasia, and this hyperplasia is also occur in response to various growth factors which are released by intact cells, intact hepatocytes, which stimulate their growth and lead to restoration of liver mass. What is pathological hyperplasia? Pathological hyperplasia occurs when there is more than normal hormonal stimuli. Then there is pathological hyperplasia. For example, in females, the uterine growth can sometimes be abnormal due to increased stimulation of estrogen. There is a critical balance between estrogen and stimulatory effect of estrogen and inhibitory effect of progesterone and pituitary hormones. All these interact in a way to maintain the growth of uterus. If this balance is disturbed and there is more than normal stimulation by estrogen, this cellular growth bypasses the normal growth mechanism, and as a result lead to the hyperplasia. Moreover, benign prostatic hyperplasia in males is also another example of pathological hyperplasia induced by male androgens. So one more example of pathological hyperplasia is hyperplasia induced by papilloma virus, which leads to skin warts and mucosal lesion. This is also hyperplasia caused by pathological conditions. So hyperplasia, the main distinction between hyperplasia and cancer is hyperplasia is the controlled growth of cell in response to some stimuli. As long as this growth stimuli is present, cell continue to divide. But when this growth stimuli is removed, hyperplasia disappears. But in cancer, even if there is no growth stimuli, cell continue to divide regardless of their growth factors, and this is basically distinction between cancer and hyperplasia. And hyperplasia is a fertile land for cancer to develop. Hyperplasia can ultimately lead to the cancer if it is not controlled, and some way control balance are lost. So this was about hyperplasia. Hyperplasia was also two types, physiological and pathological. Physiological hyperplasia was also of two types: hormonal hyperplasia, which was the hyperplasia of glandular epithelium of female breast, and compensatory hyperplasia, which was the hyperplasia of the hepatocyte after transplant surgery. And pathological hyperplasia occurs when there is more than normal hormonal stimuli. For example, it occurs in the female uterus, as well as benign prostatic hyperplasia, as well as human papilloma virus also induces hyperplasia, leading to skin warts and mucosal lesions. So next come toward atrophy. Atrophy is the shrinkage of cell size. cell size due to loss of cellular substances. In atrophy, cellular size decreases and ultimately the size of organ also decreases. So atrophy example here is when there is no muscular usage, or when a person is bedridden for a long time, his muscle atrophy due to loss of function. So decreased workload is also cause of physiological atrophy. Moreover, pathological atrophy can be due to diminished blood supply, ischemia, aging. Aging is also a physiological factor, and endocrine stimulation is lost, loss of innervation. These factors contribute to the atrophy of different organs. So atrophy is a result of when there is atrophy, the protein synthesis in the cell decreases due to reduced metabolic activity, and the protein degradation is increased by ubiquitin proteasome pathway. What happens when there is no cellular function or nutrient deficiency? Ubiquitin ligase is activated, which targets the protein to proteasome for the degradation. So atrophy occurs basically due to decreased protein synthesis and increased protein degradation in a response to deficiency of nutrients, hormonal stimuli, diminished blood supply, and decreased workload. And atrophy sometimes is associated with atrophy. Atrophy is a process of cellular eating in which cell consume its own organelles and contents to obtain energy in the time of nutrient deficiency. Next adaptation is metaplasia. Metaplasia is a type of cellular adaptation in which one adult cell type changes into another adult cell type, and this is basically by mechanism of shifting of the programming reprogramming of the progenitor cells to from one adult cell type to another cell type. For example, one cell type was initially one progenitor cell was programmed to produce columnar epithelium, but when there is metaplasia, it produces stratified epithelium instead of columnar epithelium. This is example of metaplasia. So example of metaplasia include the metaplasia of respiratory epithelium. Normal respiratory epithelium is ciliated columnar epithelium, but when a person smokes chronically, this ciliated columnar epithelium is replaced by stratified squamous epithelium. So this stratified squamous epithelium is better adapted to withstand the toxic chemicals in this cigarette smoke. So it is basically an adaptive response to better tolerate the toxic chemicals of smoking. But when there is replacement of ciliated columnar epithelium with stratified squamous epithelium, the mucus secretion or mucus function is lost, as well as ciliary clearance of particulate matter or the foreign particles from respiratory tract is also lost. So basically it harms in the normal respiratory function. Normal respiratory function is compromised. Ciliated columnar epithelium secretes mucus, as well as there is ciliary action which traps foreign particles and clear the respiratory passageway. But when it is replaced by squamous epithelial cell, this both function, mucus function and ciliary function, is lost. But this squamous epithelial cell, stratified squamous epithelial cell, is better adapted to tolerate this cigarette smoke. And metaplasia can also occur in opposite direction. For example, the esophagus, which has stratified squamous epithelium, its stratified squamous epithelium can be replaced by columnar epithelium, intestinal type of columnar epithelium, when there is a GERD or there is gastric reflux into the chronic gastric reflux into the esophagus. So it can occur in both direction. Either columnar epithelium can be replaced by stratified squamous epithelium, or stratified squamous epithelium can be replaced by columnar epithelium. And sometimes progenitor cells also reprogram and produce abnormal tissue in an abnormal place, normal tissue in an abnormal place. For example, sometimes metaplasia can lead to the formation of bone in soft tissue, which is due to metaplasia of mesenchymal cell, in which the mesenchymal cells, instead of producing a bone in normal cell, produced in some abnormal location, produce normal tissue in some abnormal locations. So these metaplastic changes of epithelium, if persistent, they may predispose to malignant transformation. So this metaplasia or metaplastic tissue is also a fertile soil for cancer to develop. So these were today's all about today's topic, in which we study about hypertrophy, atrophy, metaplasia, and hyperplasia. Thank you.